It's my pleasure today to introduce Helen Chu for the Viruses and Vaccine Seminar Series. She'll be talking about using household studies to understand respiratory virus transmission. But before we get to her talk, I'm going to do a very brief, very brief genomics update for SARS-CoV-2. The Here we are here, and so. Uh, when I spoke on this before, I think it maybe was November. It was when JN1 was first emerging. So if we look now at the kind of the entire tree here of diversity in SARS-CoV-2, we can see these like really notable breaks where there's kind of no sampled viruses here, no sampled viruses here, no sampled viruses here, no sampled viruses here. These are believed to be cases where there's a single chronic infection kind of incubating a very evolved virus, and that kind of cat and mouse game with the human immune system really pushes things to evolve antigenically. And so when those occasionally kind of stay being functional in terms of human-to-human transmission, you get these new variants that really take over. And we definitely had that initially with Omicron, BA1, BA2 lineages, again with one of the major components of the XBB recombinant, and more recently with BA.2.86 and JN1, which has really swept through the world. And so then if we look at viruses from the last 2 months, it's all of these, we can see that almost everything collected worldwide is JN1. There's a few of these XBB-derived viruses that are sticking around, but they seem to be on their way out. Interestingly, here for JN1, we saw that the initial kind of chronic infection was this BA.2.86 lineage. This looks like it started in someone in Southern Africa in spring 2022, is in this single person for, say, over a year, emerging in summer 2023. And then it actually doesn't do that well initially. It does like okay, and it competes with XBB, but is not XBB-derived lineages, but is not really taking off until you get this additional 455S mutation, which helps it to escape from antibody binding and even more, which XBB had already acquired. But this 455S on top of the BA.2.86 background really causes JN1 to take off. So this is just looking back a bit. We can see just how quickly it swept through the world, each of these red lines. And then this is basically as fast as we've seen things since maybe like BA5. It's been one of the more successful variants. And so because of that, however, like we've like bottlenecked all the diversity of SARS-CoV-2 through JN1. And so like SARS-CoV-2 is less diverse right now than it's kind of almost ever been, perhaps. And so almost everything is exactly JN1, and we're just now starting to see some variation accrue within JN1. That's going to kind of continue to happen. Right now, the most interesting thing is that spike 572 to I. You can see this pattern that we often see where if there's something that like evolution wants that is selected for, it's like happens again and again and again. And then we also have this lineage JN1.7, which is starting to take off and has accrued this 572I mutation. And if we look across diversity of viruses, we can see that JN1 is still kind of the most frequent lineage today, but JN1.7 has a bit of fitness advantage to it, maybe a 20% fitness advantage, but it's still under 10% frequency. So maybe this will continue to grow, or maybe we'll get some other 572 bearing variant that then takes off, or some other thing on top of this. And yeah, we'll see what happens. I'm gonna stop there. And now properly introduce Helen. So Helen earned her MD from Duke University. She completed her internal medicine residency at Harvard Beth Israel Deaconess Medical Center and her fellowship and MPH at University of Washington. She's now a professor in the Department of Medicine, Allergy and Infectious Diseases at UW-Medicine. Her research is focused on emerging respiratory virus infections and maternal fetal immunity against respiratory viruses. Helen has been really remarkable in running large-scale studies and from them bringing robust scientific insight. These include the Seattle flu study, SCAN, Husky coronavirus testing, and Cascadia, among others. Helen is fearless, brilliant, and determined, and I've been fortunate to call her colleague for the past 5 years. And I'm really looking forward to your talk, Helen. Go ahead.
Thanks so much, Trevor. That's such a kind introduction. The same could be said for you. It has been such a pleasure to be able to work with you. I was just going to say for those in the audience that you can go ahead and interrupt me for questions or put them in the chat, and then I'll try my best to monitor them along the way. So I'm going to go ahead and share screen. Okay, and I'm going to go ahead and get started. So the title of my talk is It's All in the Family, Using Household Studies to Understand Respiratory Virus Transmission. And I wanted to really use the household studies as a construct for really showing you what we've done over the last 7 years in Seattle to understand community transmission of respiratory viruses and to develop new methods for remote contactless surveillance. So I'll be talking about the Seattle flu study and then as well as the recent work that we've done with Cascadia. So a little bit of background. For many of you, this is a lot of review, but we know that respiratory viruses, including flu, RSV, and SARS-CoV-2, cause significant individual and societal disruption. Interestingly, there have been a series of recent perturbations that have led to shifts in the epidemiology of viral circulation. We know that the COVID-19 pandemic led to decreased circulation of seasonal viruses and then a subsequent rebound as mitigation measures were lifted. And then we also have now multiple new vaccines for SARS-CoV-2, and then in particular also for RSV, which have the potential to really shift the age distribution of cases from the very young to a slightly older age group, can and have led to viral evolution and emergence of new variants, and also potentially create an ecological niche for replacement by new viruses in the space once occupied by RSV. So this is a figure from our seattleflu.org website developed by Leah Starita and Trevor Bedford, really emphasizing this point about the circulation of all respiratory viruses being suppressed by the mitigation measures that occurred during the pandemic. And so here you can see back in 2019 and 2020, we started community-wide surveillance and found circulation of many viruses. Once the stay-at-home order was put in place, we saw virtual elimination of all circulation for approximately 2 to 3 months, followed by rebound primarily of rhinovirus and SARS-CoV-2. And then over the last 2 to 3 years, we've now seen rebound of all the other viruses with also unusual patterns of circulation, in particular of influenza and RSV. We also know that there have been many, many new advances in vaccinology in the last few years, and I wanted to really highlight the work that's been done in RSV. So this is a snapshot from PATH, which is an organization based here in Seattle, showing the vaccines that have now been in development from phase 1 through phase 3, and now market approved. And I wanted to just emphasize that in the last year, we've now had FDA approval, CDC recommendation, and rollout of 4 new products, primarily intended for older adults, age 60 plus, as well as for infants, including a maternal RSV vaccine and a long-acting monoclonal antibody that's intended to be given at birth to all infants and protect them for their first RSV season. Just a reminder for those of you who don't work on RSV, so the RSV exists in, or the surface protein of RSV exists in a pre-fusion and a post-fusion form. The pre-fusion is the form before it fuses with the host cell membrane. And we know that that the majority of neutralizing antibody is actually directed to epitopes that are only exposed on the pre-F form of RSV. And so all of the newly licensed vaccines and preventions currently target this pre-fusion form of the RSV F protein. And so the data I'll be showing to you from the last few years is really focused on understanding the serologic correlated protection against pre-F. So what can we learn in particular from household studies? And I really do love doing household studies, and I've been doing them for a long time now, both in Nepal and in the United States. And I particularly like household studies because I feel like they provide us an epidemiologically rich source of information that help us understand viral transmission dynamics. For those of you who have young families, you know that households are a site of intense exposure. We can use households to really track community incidents across age groups, particularly in those who are in the 18 to 49 group who don't have time to go seek care for every respiratory illness. We can calculate secondary attack rates, serial intervals, generation times. we can better understand serologic correlates of protection, and then use household studies in particular to study the phenomenon of viral interference, where having one viral infection potentially protects you in the subsequent period from a second infection. Households, because of the high rates of transmission, also provide opportunities to study interventions, in particular, the use of diagnostics, MPI, such as hand washing or masking, and then the use of antivirals. And these household studies can really help us identify effective interventions that reduce transmission and allow us to test out fully remote early diagnosis and antiviral treatment trials. So the factors that we know are associated with household transmission of viruses include some household level factors as well as individual level factors. And so here at the top, what you can see is that the factors that have been shown across many studies include the density of the household, so household crowding, behaviors such as sharing a bathroom or eating space, the ventilation in the family, in the household, and having young children in daycare and school. Those all impact risk of household transmission. We also know that there are certain characteristics of the index case, such as the age, the symptom status, and the viral load. And then factors associated with the secondary cases, including age and serologic status, which impact their risk of acquisition. And then really what we want to use households in particular is to understand some of the infectious disease epi associated with new and emerging pathogens, including the incubation period between when you have the infection and symptom onset. And in particular, the serial interval, which is the time period between when the first person has symptoms and the second person has symptoms in the household. And what we know now from a lot of the COVID studies that have been done is that this information that we've gained from these early studies that were done in China and in Europe and the United States have really helped us understand these the dynamics of household transmission, in particular, the risk of asymptomatic transmission from the first case in households that have really informed public health policies in these countries. Okay, so in this talk, I wanted to go over two studies. One of them is the Seattle flu study and how we use the Seattle flu study to really develop a framework for remote surveillance for respiratory viruses in a community setting, and how we tested out a fully remote antiviral treatment approach for influenza. And then I wanted to follow this up with some early data from the Cascadia study, which is a study that we've been doing for the last 3 years to characterize the incidence and risk factors for viral infections across the age spectrum, and then to use genomics to understand household transmission of respiratory viruses. Okay. So the work has been led by 4 individuals. Jessica Hyman and Annie Emanuels were research coordinators in the lab during the period of the Seattle flu study. And Coleraine Freifeld and Sarah Cox are graduate students and postdocs in the lab who've done a lot of the analysis for the Cascadia study. And really, what I'll be presenting is all of their work. So I wanted to start first with the Seattle flu study. So many of you know this, but we started the Seattle flu study in 2018. At the time, it had been 100 years since the 1918 pandemic, and we felt that another pandemic was inevitable. And that current surveillance strategies in the United States are really not designed to detect novel pathogens. In particular, they were focused on clinical surveillance and hospital-based surveillance, and did not provide the opportunity to understand what was happening in the community, where we thought that viruses were likely to be detected first, and early detection could potentially lead to early interruption of spread. See, the objective of the Seattle flu study was to develop a pandemic surveillance platform that could be used to identify new pathogens and to develop strategies for rapid containment before the months to years it would take for a vaccine to be developed. And so one of the key tenets of the Seattle flu study was to expand testing to outside of the hospital. And we felt like hospital samples were a good comparison group. And what we wanted to do was to really understand what was happening in the community. So we went ahead and expanded to schools, universities, and daycares to congregate living settings, in particular the homeless shelters that were in Seattle, and in public spaces and workplaces, such as under the Space Needle, the light rail station, on a university campus, and in places like Costco, and places where people, healthy working adults would be found with respiratory illness and could potentially lead to transmission. And then the other piece of that is we really wanted to work within households, and households provided us this opportunity to do all the things that I talked about earlier, and to really try out a process of remote contactless surveillance for viruses that could be used in the future. So between 2019 and 2020, we developed a remote household observation study. And so between November 2019 and April 2020, we enrolled in partnership with Seattle Public Schools, households with young children. We recruited them and then remotely enrolled them and monitored them for symptoms of respiratory illness. We did this all via phone call and consents through REDCap that were virtual. And they were mailed home swab kits with instructions for use. In the event of an acute respiratory illness, they were instructed to self-collect a swab and then mail the sample back to the lab. And then we asked them to track their symptoms either through text messages or emails. And then importantly, they were allowed to view their results on a study website. So here was our study website, and it was essentially a way for people to enroll in our studies and to be able to do all of their enrollment and consent fully remotely. And this was the instruction kit. It was intended to be something where somebody could take a look at this instruction manual and really collect their samples by themselves. And this was back at a time in 2018, 2019, when really people were not self-testing for respiratory viruses. So this was quite controversial. But we were able to do this successfully for the household study and also for several other studies, and show that people were able to collect the samples themselves, to package it appropriately, and then to send it back to us, and that the samples were then able to be successfully tested. So the samples were then brought into the lab, into Leah Starita's lab at the Bratman Beatty Institute. And then we used a multiplex assay for many respiratory viruses, as well as bacterial pathogens, that we used to test these thousands of samples that came in. And in particular, we wanted to test for the traditional respiratory viruses, but we wanted to include a primer for influenza A that was not H3N2 or H1N1 specific, because we wanted to capture novel influenza, such as an H5 or an H7. And so the idea was that as samples were coming in, we would be testing them for all of these influenzas, and hopefully would be able to use this to detect novel influenza strains as they emerged in the population. And then importantly, individuals who enrolled in our study were able to get a barcode with their swabs, and then they would enter their swab onto the study website and then see the results. And initially, we were allowed to share results for all respiratory pathogens. And so individuals who were part of this, the household study, were able to go online and see, for example, whether or not they had human coronavirus or RSV. And that was, I think, a nice incentive for participation. So this was really a feasibility study. We enrolled 350 households actually quite rapidly. A lot of people were interested in participating. 303 completed at least one symptom log, and we were monitoring 1,100 individuals weekly for symptoms. And importantly, we were able to monitor age groups that traditionally did not seek much medical care. So this was the 5 to 17s, and then the 18 to 64s, which really made up the majority of those who were enrolled in our studies. We found that 213 households successfully submitted at least one swab with 678 illness episodes. And then of these 678, 565 completed all of their testing, and then also sent us all of their illness surveys for us to do the analysis. The first thing that we observed was that respiratory viral patterns in the households really recapitulated community epidemiology. That what we found in the households was very similar to what we saw from our community surveillance from Seattle Flu Study across all of our other studies and across the clinical data that was coming in from the hospitals. And so here at the top, you can see the viruses that were circulating as part of all of our other surveillance modalities. And then the household study, which really was very similar to this in terms of the seasonal patterns of the viruses in these select households. So household studies are a good way to be able to understand what is happening in the community at a much smaller scale. The other thing that we found was that there was a very high proportion of virus-positive illness episodes in households with young children. So 35% of these illness episodes tested positive for at least one non-influenza respiratory viruses. And 4 cases of SARS-CoV-2 were detected among 3 households before April 1st of 2020. And if you remember, one of these cases was the first case of community transmission in the United States that Trevor and Leah identified and sequenced. So the risk factors for detection of respiratory viruses, including the presence of a young child under the age of 5 and an adult age 18 to 49 in the household. And we did not find an effect of other socioeconomic indicators or household density. We had a fairly homogeneous population in our study. It was mostly white or Asian and on the wealthy side. So it definitely did not give us the distribution to really look at some of these other factors that have been associated in past studies. One of the things that we were able to do is we were able to capture very, very detailed symptom data about all of these illness episodes, stratified by age group, and then also by pathogen. And you can see just as the color gets darker, it's more commonly detected with that virus in that particular age group. So RSV in under age 5 was associated with a lot of cough and runny nose in this population. And then what you can see here, I think most remarkably, is that as you get older, you are much, much less likely to have fever with your illness episode. So using this ILI criteria for detection of respiratory viruses, in particular influenza, is not something that necessarily holds in the older age groups. And it's quite useful for younger children, but probably for older adults, we need a broader symptom criteria for respiratory virus detection. The other thing we observed in the household studies was that there was quite a burden of respiratory illness in this healthy working adult population. So 57% of the illness episodes in this population had a virus detected. We had a lot of human coronavirus, rhinovirus, human metapneumovirus, and RSV in these adults. What we noticed, though, was that very few of the people with these respiratory viruses sought medical care. So very low proportions, 12% of individuals with a symptomatic respiratory illness sought care, but 25% were absent from work and school. So though these viruses cause symptomatic respiratory illness, they did not lead to care seeking and then would not have been captured by sort of traditional surveillance mechanisms that really require you to come to the doctor, come to the hospital and get tested. But it did still lead to a lot of a high burden of sort of morbidity with work and school absenteeism, particularly for certain viruses that we know cause that, including RSV. We also looked at secondary attack rates of these viruses, and we found that overall, this was pretty similar to what has been seen with other types of household studies, that secondary attack rates, particularly for RSV, around 30%, which is expected. And then we found the same for human metapneumovirus and rhinovirus. I think some of this data actually will be very useful as we think about all of the new vaccines that are coming down the pipeline, particularly those that are directed against human metapneumovirus, because it gives us a good understanding of what the burden of disease of all of these other non-RSV, non-flu viruses are in households. Excuse me. And then one of the questions that household studies really provide us an opportunity to answer is to address this question of viral interference. So the phenomenon of viral interference is that once you have a primary viral infection, it leads to an increase in your innate immune response and subsequently protects you from acquisition of a second infection. And this has been seen certainly in animal studies, and then also in some epidemiologic studies where a prior rhinovirus infection appears to protect against a subsequent influenza infection. And this is an important question, particularly in this time, because as we start eliminating some of these viruses in very young children, this question of viral interference or replacement does come into play. So we use the household study to try to address this question, to look at whether or not a primary infection with one virus was protective against acquisition of another virus in the subsequent 90-day period. And unfortunately, we did not find it to be the case. And so we actually found the effect to be in the other direction, that having a primary virus, in particular with rhinovirus, with parainfluenza, and with RSV, were associated with an increased risk of a subsequent viral infection in the same individual over this 90-day period. And there are epidemiologic studies that have shown this as well. I think these studies are very challenging to do because of the seasonality of viruses. So all viruses seem to circulate at the exact same time, and it's very difficult to adjust for co-circulation of these viruses. But we did not find actually this effect that has been seen in some of these early animal studies of protection against a subsequent infection in this post-viral period. Okay, so now moving on from the epidemiology to the clinical trials. So as part of Seattle flu study, excuse me, I'm still getting over something from a trip to India. As part of Seattle flu study, one of the challenges that we really wanted to address was the idea of being able to identify pathogens early enough to be able to intervene to stop transmission. If you remember from before the pandemic, when we used to treat for influenza, one of the main things was that people just waited too long to come to seek care. And so they couldn't receive their oseltamivir or their baloxavir early enough within that 72-hour window for it to be effective in reducing symptoms. And so one of the things we wanted to see is, could you then move this completely to the household and thereby shorten the time to detection of the virus and treatment of the virus so as to stop within household transmission and then potentially community transmission. So we started this study again in 2019, and the idea was to really provide proof of concept. So again, the households were enrolled. This was a subset of the surveillance study that we had done that I just talked about. We did weekly text messaging for illness weekly text message surveillance for illness episodes. And then importantly, if people had symptoms of a respiratory illness, we asked them to self-test at home for influenza using a home-based test. And this was a test that was developed by a company called Ellume, based in Australia, where it was an antigen-based test, and it emitted a fluorescence signal that then Bluetooth to your phone and would provide you a result of whether or not your test was positive or negative. And would also send an email to you as well as to your provider with that result. So based on the self-test, if people tested positive for influenza, we asked them to consult a telehealth provider, and the provider that we used was called 98.6, and they're based here in Seattle. And we worked with them to be able to then assess this individual, to use the test result to make a clinical decision. And then we have a prescription sent to the pharmacy, the UW Investigational Drug Pharmacy, for baloxavir, which is a 1-dose antiviral, which was then delivered to the house within 2 hours to be able to treat people for influenza. We asked individuals to also continue to collect swabs to monitor first for concordance with the home-based test, and then also for potentially emergence of baloxavir resistance, so that we could see if this dose of baloxavir could potentially lead to resistance, as had been seen in some clinical trials earlier. So this is a schematic of how this test worked. So essentially, people would use this swab. They would swab their nose. They would put it into the kit. The kit would then read the result and then emit the signal to the phone. And then the phone would then essentially send you a message and say, your flu test was positive or negative. So we conducted this study starting in November 2019. It was intended as a 2-year study, and it was really the outcome was household transmission, interruption of household transmission. However, unfortunately, very early on in the conduct of the study, the Washington State Department of Health requested us to stop the study. And the reason why was because they were concerned that people were not able to test themselves for influenza, and that using a research test for self-testing was going to lead to inappropriate diagnosis and treatment for influenza. So the results were only from these 3 to 4 months of surveillance. So we enrolled, in this time period, we enrolled 150 households, 481 individuals, and then 302 were eligible for baloxavir. We conducted 58 home flu tests during the surveillance period, and we had 12 positive results. Here on the right, what you can see is the sensitivity and the specificity of the home flu test as compared to the molecular test that was done in the lab with the samples they sent in. And I thought it was actually pretty good, a sensitivity in the 70s overall, and a specificity of 96%, and basically as good as you can get for a home antigen test. We found that people were able to access telehealth providers. We conducted 11 telehealth visits for home flu test positive episodes. 8 of these were Baloxavir eligible, and then all of them received home deliveries. We delivered the Baloxavir in a median of 1.6 hours from time of diagnosis and telehealth provider prescription. And then 88% of deliveries occurred within 3 hours of Baloxavir prescription. We were able to deliver the antivirals 25% within 24 hours of symptom onset, 37% between 24 and 30, and then the last 37% between 30 and 48 hours. And we really, if it had been more than 48 hours, people weren't allowed to enroll in the study. So we really wanted to see if we could capture them early. One of the things that we were able to assess in the study was whether or not text or email was better at getting people to respond. And this was really very early in early days before we now routinely use text messaging for symptom-based surveillance. But this is part of the reason why we now do that. It's because we found people were much more likely to respond to text quickly compared to email messages for their weekly symptom surveys. We had remarkably high compliance over the course of this study period. We found that something like 95% of people completed their weekly surveys, but that they really liked text messaging. And so after this study, we really fully pivoted to text messaging as a way to conduct remote surveillance for symptomatic illness. And then as part of this, we were also able to see how people would respond to a test result. So among the 47 participants who completed a home test and follow-up questionnaire, we asked them a couple of questions about what they would do with that positive test result. So 94% reported that their symptoms and positive tests would lead to minimizing contact with others, compared to 78% if they had symptoms but they didn't have a test result. And 89% with symptoms and a positive test would miss work or school, compared to 59% for symptoms without a test result. And so having a positive test result for influenza changes your behavior, or theoretically changes your behavior, and you would minimize contact with others and be more likely to be absent from work and school. The people who participated in our study thought that the test was easy to use, the app was easy to use, the results were easy to read, and they felt confident in using this test. So this is one of the very early studies that really showed us that using home-based testing and treatment for a respiratory virus is possible and is something that could be implemented in the cases of a pandemic. So from these studies, we concluded that there is a high burden of symptomatic respiratory viral illness in healthy adult populations with associated work and school absenteeism, but not as much healthcare seeking. We were able to assess this concept of viral interference in the community using the household studies. And we were able to do studies that demonstrated proof of concept for fully remote home self-test, clinician diagnosis, and treatment for influenza. And so now I'm going to move on to the work that we've done now in the post-pandemic period through the Cascadia study. And this is really work that has spanned this time period, in particular of after mitigation measures were lifted and RSV has resurged and led to cases in populations that are not traditionally at high risk for hospitalization, including older children. And then also, this is a very unique time period right before the start of vaccines, and then these monoclonal antibodies that are now, in this past few months, been implemented for use in young children. So it provides a nice baseline for assessment of all of these interventions that are now ramping up in the community. So the Cascadia study, a brief overview. So it's a prospective observational study, 3,500 children and adults in Washington, Oregon. People need to be 6 months to 49 years to enroll. And the primary objective of the study is vaccine effectiveness of SARS-CoV-2 vaccines in pediatric and adult populations in a community setting. And that study has been published and is cited here in the reference, but I'll really be showing the data that has not yet been published. So we enrolled them and followed them weekly for respiratory symptoms. We collected a nasal swab in all individuals, those with and without respiratory illness. And excuse me, all swabs have been tested for flu, RSV, and SARS-CoV-2, again, in the BBI lab, led by Leah Starita. Symptomatic swabs were also then tested for 26 pathogens, and then we sequenced all positive samples. We also collected serum samples in all individuals every 6 months, and then for a subset, day 30 post-infection and post-vaccination. This is the schematic of the overall study design. Individuals enrolled in the study, we collected a lot of socio-demographic data about their vaccines, their household composition, their health status, long COVID symptoms. And they completed many surveys, and then weekly text message-based surveillance for respiratory illness, and then collected a nasal swab every single week. These are some of the pieces of the kit that we use for our study. This was developed by the BBI logistics team led by Zach Acker. On the top left is a picture of a swab kit that we send to the individuals enrolled in our study. On the top right are the components of the kit for self-collection and return. On the bottom left is a swab that we use, and you can tell that it's very small. It's called a rhinostic swab. It's plastic. And it's collected without and transported dry. And so it saves us quite a bit of money to use this type of swab. And on the bottom right are these stickers that we developed. These are Pacific Northwest animals. And because we primarily focus on enrolling households with young children, we needed a way to differentiate the swabs that came from all of the different individuals, because they're collecting every single week. And if you have young children, you know that it's very hard to figure out to keep everything straight, especially if you're busy or you have a respiratory illness. So we asked people to identify an avatar for themselves that they would use for the duration of the study. And so people would select from all of these and decide what sticker they would want on their swab kit. And then that sticker would be put here on the kit so that we wouldn't mix up the samples. I think the most popular ones are the fox and I can't remember if it was the banana slug, but those were these have been very successful in our study. If you want to talk more about doing this type of work, I'm happy to talk to you. But we found this to be really nice for engaging young children and for very high compliance in the Cascadia study. So overall, we enrolled over 3,500 individuals. We've now followed them for 2 years, and approximately 250 were less than 4, 844 between the ages of 5 and 11, and 644 between 12 and 17, and then the rest of them were adults. We found high levels of influenza and RSV in our population over the course of the 2 years, and I will really be focusing the results on this first RSV season, although the final analysis will probably include some later results as well. And then this is just to demonstrate that we also, or rather Leah's lab, tested all of the samples that were symptomatic for all of these other pathogens. And so this provides us a real opportunity to understand the burden of all of these other pathogens in a community-based setting in this post-pandemic period, including metapneumovirus and parainfluenza virus and the human coronaviruses. So first, I wanted to just share the baseline characteristics of households and individuals with and without RSV infection. And remarkably, they weren't actually that different, other than the fact that people who are RSV positive were more likely to live in a household with children under 5 who are attending daycare, which we know is a clear risk factor for RSV infection. We also found that many more cases of RSV in children as compared to adults, and that we found that the majority of the cases of RSV were actually in children between the ages of 6 months and 12 years. When we look at incidence of RSV in this first post-pandemic period, we found that incidence among all participants was shown here in the black. But then when we stratify by age group, what you can see is that the cases occurred the most and then also the earliest in the 6-month to 1-year-old group, as well as in the 2 to 4-year-old group. And so really, we saw RSV show up first in the very young. And then in the community, what we saw was that it showed up in the older children, the 5 to 12-year-olds, and then the adults, suggesting that the RSV outbreaks have really been starting in these in places where very young children are, which is probably the daycares. We also have looked at symptoms in all of these different age groups with their RSV illness episodes, and found that cough, rhinorrhea, and sore throat were commonly detected among all age groups. But then importantly, that we did not see fever here in the older adults or the teenagers who had RSV infection, which is similar to what we found in our earlier household studies. This is a snapshot that shows sort of the granularity of the data collection that from Cascadia that we've been able to do over the past 2 years. So each line represents one individual. And really what we've zoomed in here is just showing you the 6-month to 1-year-old group, and then the 2 to 4-year-old groups who've had RSV. And so each black dot represents a swab that was taken from them over their course of participation in the study. And then the red dot represents the RSV positive illness episode. And then if you remember, everybody had a blood collection that was at enrollment, so somewhere along here at the very beginning of their study. And then they had a subsequent RSV illness episode while they were under surveillance over the course of that 2-year study. One of the first questions we wanted to answer was really around the idea of is this viral load associated with age or symptom status? And we found that to be the case for both pediatric and adult populations, that people who are symptomatic had a higher or a lower CT value, a higher viral load compared to those who were asymptomatic, had RSV in our study. We had a very broad symptom criteria, but for the purposes of this analysis, we restricted to symptomatic to cough and or rhinorrhea in that time period, and then found that people who were symptomatic, both the children and the adults, had higher viral loads, but the adult and pediatric viral loads are actually not particularly different as long as they had symptoms. The other question we really wanted to answer in the context of this study was what is the relationship of baseline serum antibody titers to subsequent risk of infection? So what we did was we took all of the bloods that were collected from the children and the adults in the study, and we did a prefusion ELISA using the MSD assay on all of the samples. And then for the pediatric samples, we also did an RSV neutralizing antibody assay in the UW clinical virology lab with Alex Gruninger. And what we found, first of all, is that the age distribution of the pre-F antibody varied according to age, or the titers of RSV pre-F varied according to age. The very young had much lower titers. The 2 to 4-year-olds also had fairly low titers, and then as children got older, 5 to 12s and 13 to 50s, they all their pre-F antibody titers seem to have reached a plateau. What was also remarkable is just sort of the distribution of these antibody titers, particularly among the very young. The next thing we wanted to do was to understand whether or not there was a difference in their baseline antibody titers by whether or not they subsequently had an RSV infection event in the follow-up period. So does baseline higher titer protect you against a subsequent RSV infection? And we found this to be the case in the young children. So in the 6-month to 1-year-olds, that those who are RSV positive had a lower baseline antibody titer compared to those who are RSV negative. And the same was seen in the 2 to 4-year-olds. As you got into the adolescents and the adults, though, this effect went away. And so when we looked at a hazard ratio of the association between the baseline pre-F antibody and the subsequent risk of RSV infection, we found that that overall that a higher antibody titer at baseline was protective against a subsequent RSV infection, and that in particular this was seen for the pediatric participants. And so, the pre-F ELISA is actually not a standard assay that's used in clinical trials. And it's something that's more recently been developed, in particular in the setting of the use of all of these pre-F-based vaccines for RSV. So the other question we wanted to answer was, what is the relationship between what is considered the gold standard, which is the RSV neutralizing antibody assay, and the pre-F antibody? And we found that these results were very highly correlated, that the pre-F antibody titers were predictive of neutralizing antibody titers among pediatric participants. So if you were to do a study where you had you had limited samples and limited time and money, that pre-F antibody is a reasonable choice rather than doing a neutralizing antibody on all of the samples, which is very, very time intensive and costly compared to doing an ELISA, a multiplex ELISA. So finally, in the last few minutes, I wanted to share some of the data that we have on very early data on using genomics to understand household transmission. And so in our study, because we really prioritized enrollment of families, of households with children, the majority of the households that were enrolled were multi-member households. So we had 3,238 individuals enrolled, and we first excluded all of the single person households, which wasn't that many. It was about 178. And then we identified those that had an RSV episode. And so that was 96 of the 1,010 households. We divided them into those who had just one RSV case, so no evidence of onward transmission, and compared to those who had at least 2 RSV cases, and then divided these into 2 or 3 groups. Those who had likely transmission, which we considered a second event within 14 days. Those who were unlikely to have transmitted, so who had a transmission event more than 14 days, and then those who had co-primary cases, so 2 people who had RSV detected on the exact same day. And so the first thing we did was we just looked at the factors that were associated with and without household secondary transmission. And again, we didn't find a whole lot of difference between the 2 groups. We found that households that did have secondary transmission, 50% of them had a child under 5, as opposed to 44% of those who did not have secondary transmission. We also didn't find much of a difference in symptom status, of the index cases in the households that had secondary transmission. 95% had symptoms as compared to 88% in those without secondary transmission. And then we also found that overall that the age distribution was not particularly different. That really the driver of household transmission, it appears to be these young children, so those who are under 12 years of age. And this is a little bit different from studies that have been done in the past. The most recent studies have been done in Kenya and have really shown that school-age children are the drivers of transmission. And what we find here is that it's actually both the school-age children as well as those who are under 5 years of age, who are probably those who are enrolled in daycare. And then what we wanted to do was to use whole genome sequencing to understand whether or not this 14-day cutoff was appropriate. This is a sort of a question in the field that we talk about a lot, is what is the right cutoff between, what is the right sort of serial interval between the cases, and what is sort of the appropriate cutoff for assessment of transmission in a household. And so this is early data, but what we did was we used whole genome sequencing to sequence all of the samples collected from households. And we generated this phylogenetic tree that shows first a diversity of the lineages that were represented during this time period of surveillance. And then what we did was we looked at the mean pairwise difference of the sequence samples within the same household. And this was work that was led by Pavitra Roy-Chowdhury. And we found that for cases of where the 2 cases in the household were within 14 days of each other, the mean pairwise distance between the sequences was 2.85. And for those where the 2 cases were found more than 14 days apart, the mean pairwise distance was 136. suggesting that this 14-day cutoff is probably appropriate for epi analysis based on the genomics that showed that if you were more than 14 days apart, you probably had a different strain that was that was separately being introduced into the household as opposed to within household transmission. That was reassuring to us to see that the cutoff that we had picked seemed appropriate. So in conclusion, in the post-pandemic period, RSV incidence was highest in young children, and particularly among those in daycare. We found that higher RSV antibody prior to infection is protective against subsequent RSV infection in children in a community setting. This is important because a lot of this data that has been previously generated has been from clinical trials and in healthcare settings. Household transmission of RSV occurs frequently in households with young children and in those with symptomatic primary infection. And that secondary cases that occur more than 14 days after the index may be more likely due to independent introductions than within household transmission based on the sequencing data. So acknowledging all of the Seattle flu study investigators, including many people on this call, in particular Leah and Trevor, who've led so much of the work in this space. And then the Cascadia study investigators, this is just a very, very small subset of all the people who've worked on this massive study over the last 3 years. And then, of course, all of the study volunteers who have participated and given us their time and their noses for the last 7 years and really did all of this incredible work to give us all of the data that I'm presenting today. So thank you. Happy to take questions.
Yeah, thanks so much, Helen. So yeah, please put questions in chat or just raise your hand and or just speak and we'll take the questions.
Hi, Helen.
Hi.
Thank you. That was fantastic talk, and I'm super cool to hear about the Cascadia study. I wanted to ask if you were able to assess anything about the people in the household who got those secondary infections. It seems like probably the numbers are pretty low at this point, but whether you have plans to try to assess whether some people are being protected for some reason or not from that.
Yeah, that's the idea, is to go and look at the secondary cases and look at their baseline antibody titers. The problem with the study, as with all studies, is that you can't sample everybody continuously. But it would be great if we had a titer right at the time of their infection, but we will use methods to be able to extrapolate what their titer might have been at the time of their infection to see if it was protective against infection. And of course, we'll look at characteristics of the index, like the viral load and things like that, to see if that impacts transmission to household members.
Helen, that was great. Yeah, there was a lot of data. I was really interested and reassured by what you showed about the MSD assay and the comparison with neutralizing titers for RSV. And what is your sense of being able to use something like MSD to monitor like the immune status of the population through time? And like if you have a serum sampling at the end of the season, do you see a rise in RSV titers in those who've been infected?
Yeah, as in how reliable is the quantitative component of MSD? I don't know that yet. We are starting to get that data in now because we have 2 years of MSD in every single pediatric and adult participant, and then we know when they had their infection events. But there is quite a distribution in their titers, at least at their very first time point, but we haven't looked yet over time to see whether or not what the full change is, or if there's a change in their MSD over the course of infection. We also haven't looked at it for neutralizing antibody yet. But we'll have that data. And Chidozi, the PhD student in my lab, is analyzing that right now. There's a question in the chat about how confident are you that PanFlu APCR will pick up a new flu strain, including avian flu? It's supposed to pick up H5 and H7. I think Trevor may be able to answer that question a little bit better than I can. I think they are proprietary primers to Thermo Fisher, so we don't exactly know what they they detect, but Leah's, oh, actually Leah's on the call too. So she might be able to know if there's been testing in the lab for H5 and H7.
I don't know for this specific PCR primers, but like you should be able to have primers that will work across across flu A. That's in both birds and humans. But yeah, I don't know these specifics.
Yeah, Thermo says they should be able to detect this. I have not tried this, but it's kind of legal to have those in the lab. But yeah, and we don't know the sequence because we are.
So I had one very related to what Lauren and Cecile were asking about, of like just thinking about this a little more almost like conceptually, that we have 50% of, let's say we have a situation where we have 50% secondary attack rate in the household. And then like fundamentally, how deterministic is this? Like we think it's related to immunity and so forth. And then if you look at baseline titers, you might see that people that have higher titers are less likely to be infected than others. But like how, like if we had a perfect readout of immunity, would it become completely deterministic and we would just know that these people would be infected and these people wouldn't? Or how much like space is there for actual stochasticity as well? And thinking about the secondary attack rate and like, yeah, how even if you had a very good readout of immunity, like how predictive do you think it would be of secondary attack rate?
I don't think it would be perfect because there's also we're looking at infection, not lower tract disease, and I feel like most of the data shows that a higher neutralizing antibody titer is protective against LRTI, lower tract infection. And we don't have the numbers of cases of LRTI to really sort of understand that. But I think that a lot of the things like viral load, number of contacts, repeated contacts, intensity of exposure, those can overcome your antibody titers and you can acquire infection. Whether or not you get sick, I think is really determined by your serum neutralizing antibody titer. It would be nice if we had mucosal sampling, but that is one of the drawbacks of using the rhinostic swab is that because it's a dry swab, we're not able to really look at IgA.
Thank you. I think we have time for one last question. Shifting gears a little bit, Isabel asks, how likely is it that environment surveillance, e.g. sewage, will be reliable as household surveillance?
I think it's pretty good. We've done a couple of these types of studies now in elementary schools and in homeless shelters, and found that the environmental surveillance really is quite matches what we find in people's noses at the same time. So, but I'm happy to share that data with you. So
cool. Thank you, Helen. Yeah, I'm sorry. There's a couple more questions, but I think we should probably end to be on time. Yeah, thank you all for attending. Thank you, Helen, and we'll announce in terms of the upcoming seminars soon. And thank you all.